We will use the DNA IQ system to isolate our own genomic DNA. One thing that's needed is a sterile foam pad. So you can see this foam pad is sterile and has never been touched by human hands before. So we're going to open it very carefully, touch it by the end, and pull it out. And we're going to swab the inside of our cheek, making sure that we have not eaten anything in the last 15 minutes to allow our saliva to wash any food debris out of our mouth. Because just in the same way that we have DNA in our cells, if you eat chicken, there's chicken DNA in chicken cells. If you have spinach, there's spinach DNA in spinach cells. So you want to make sure that your mouth is free of food debris. So you're going to rotate this foam pad around your mouth like this, very vigorously, like brushing your teeth. Now you have a lot of your cells collected on the foam pad, and you want to put it into a tube. But you're going to need to spin this tube, and you can't do it with the stick like this. So you're going to have to break it off, and you want to raise the foam pad slightly in there before breaking it off. And now you can close the tube like this. So then we're going to add 250 mils of prepared lysis buffer, and our lysis buffer will have dithiothreatol added to it, and this will help it break open the cells. After we add our prepared lysis buffer to it, we're going to vortex it and put it at 95 degrees Celsius for 30 minutes to allow our cells to break open. So now we've had our DNA sample in our pad at 95 degrees for the 30-minute time period. And we're going to remove it from 95 degrees and we're going to vortex it. Then we want to remove all the solution from it that we can. Squeezing the pad in the process to get all the liquid that we can out. And put that into a new tube. If you feel that you have a significant amount of liquid left, you can centrifuge. You need to have a balanced tube in there. And you can set it to spin for 1 minute. After you're finished centrifuging, you want to make sure that you are here to release the lid, collect your sample, and get any extra liquid that has come to the bottom of the tube because of the centrifugation. If you try to wait, then you will not be able to collect it because the pad will soak it back up. At this point, you want to add any extra liquid to your tube now containing your DNA. You're going to then add 7 microliters of the resin, making sure the resin is well mixed, 7 microliters of the resin to your DNA sample. So what's happening now is the guanidine that's in the lysis buffer is allowing a salt bridge to be made with the iron core beads of the resin, and it's making a salt bridge to your genomic DNA in the sample, to any DNA that's in the sample. So after you have done this, you're allowing it to mix and vortexing it for just 5 seconds to make sure it's mixed. And then you're going to put it in the magnetic stand. And just like you saw earlier, it's all separating off to the side. So all of those magnetic beads are now to the magnet, which is in the center, the magnet side of the tube, and all of your DNA is now being carried along with those magnetic beads. So then you can open the lid and you can remove the sample buffer that is no longer carrying your DNA because your DNA is all fixed to those beads in the side. So now the tube is empty, but those beads are still fixed to the side of the tube. So now we're going to wash it with that extra 100 microliters of lysis buffer. Remember in the beginning, you made 350 microliters of lysis buffer. We used 250 of that 350 microliters of lysis buffer to put in with your DNA in the first place. And now we're going to use the extra 100 microliters that you have not yet used. So you're going to put that 100 microliters of lysis buffer in that has the DTT in it. And you're going to vortex. And again, put it in the magnetic separation stand. And again, you can see all of those beads snap over to the side and the buffer is in there. So what we're doing is we're washing off the beads. So again, removing all the buffer. And then we're going to add our wash buffer. So we have our 1X wash buffer, and we're going to add 100 microliters of the wash buffer. As before, vortex. And that wash buffer is then washing off those beads to make sure we're getting rid of RNA, proteins, things like that, which might be adhering to the DNA. As before, putting it back in the stand, letting those beads snap over to the side, and removing the buffer. And you're actually going to do this procedure 3 times. You're going to wash it with the wash buffer 3 times. Each time you do it, you're adding 100 microliters of the wash buffer. You are removing from the stand to vortex, putting it back in the stand, and discarding the wash buffer in there. When you have finished doing this, then you're going to open the top of it and just let it air dry. So after you've done it the 3rd time with the wash buffer, you're going to open the top and just let that air dry. And then you're going to add 100 microliters of the elution buffer. So the elution buffer is again in a pre-packaged container that looks like this. The difference between the lysis buffer and the wash buffer and the elution buffer, the lysis buffer and the wash buffers both contain guanidine. So this maintains that salt bridge between those beads and the DNA. The elution buffer does not contain guanidine. And so because it does not contain guanidine, all of the DNA, once you add the elution buffer, the DNA will come away because there is no longer a salt bridge connecting it to the beads. So the DNA will then be in solution. So then, after you have done this 3 times and snap those beads over to the side, when there is elution buffer in here, now your DNA will be in the solution instead of attached onto the beads, and the beads can then be simply be a waste product. So then you're going to take your sample of your after you add the elution buffer, you want to make sure that you have it at 65 degrees. So you're going to be putting it at 65 degrees for 5 minutes with the elution buffer in there to allow time for that elution to take place. And again, vortexing, putting in the stand to be able to get your DNA, which will now be in the supernatant instead of in the beads. And then you can take your sample of your DNA and put it into a PCR reaction and amplify your PCR with 3 different sets of primers. So we'll be multiplexing in a PCR reaction. We'll be amplifying 3 different places on the genomic DNA at the same time. So now we're ready to do our PCR reaction. And for our PCR reaction, we have these small tubes. This is the size of the tube we've previously been using, and this is the size of our PCR reaction tube. This is good for PCRs of 25 or maybe 50 microliters, but you don't want to do too much in a tube like this. And the PCR machine has a special block in it designed to fit these much, much smaller tubes. Some PCR machines are designed to be done with 10 microliters, and with some you can even do samples as small as 1 microliter, depending on limitations in the amount of DNA that you have. So you want to make sure you have your protocol in hand when you are doing this, and to your tube, you're going to add 16.5 microliters of sterile water. You want the water to have been autoclaved so that if there's any contamination in it, it's going to kill any contamination. You want to use water fresh from the distilled, deionized machine that puts out very, very clean water. We have a distiller upstairs. And then you autoclave it to make sure it's very clean and stays in the bottles to avoid contamination. We also have our 10x buffer. So we have for our kit, we just have a special tube of 10x buffer, and we have specific what's called CTT primer pair mixes. Make sure if you don't understand what CTT are, make sure you watch the video where I explain the theory of what we're doing in this PCR reaction. But we're doing a multiplex in this reaction, so we're going to be amplifying the DNA at 3 different locations. At the location on the DNA called CSF1PO, at TPOX, and THO1, so CTT. So there are primer pairs in here for all 3 different sites on the DNA. In addition to the primer pairs and in the buffer, there's tris to give it a good buffer. There's DNTPs. There's a little bit of salt. Everything that you need to have a good PCR reaction happen. And then you want to have a positive control and a negative control. So for our positive control, we can be doing K562 DNA. This is human DNA that will amplify at the same locations that same area, just same general area locations that your or my DNA would amplify at. It will have different sizes than your or my DNA will in general, but it will be the same location on the DNA that's being amplified. So you're going to put in one tube some K562 DNA to make a positive control. So generally, whenever you're doing a PCR, you have at least 3 different tubes minimum. You have a positive control with everything just the same that you're putting into your PCR reaction, but you have some DNA that you know will work. And you have a negative control where instead of putting in any DNA to be amplified at that location, you're just substituting some water in. This helps measure contamination. And measuring contamination is a very, very important concept for forensic science labs. Forensic science labs go to great extents to avoid any type of contamination in the lab. They're using gloves when they're touching anything. Generally, they're wearing a face mask when they're working with samples to avoid, breathing or coughing or spitting when they talk, any of their own DNA onto the surface where they're working. So they will not even talk when they are working with a sample. If they need to say something to somebody, they will stop what they're doing. They'll make sure everything is closed up, and then they will turn around and never talk over their samples. They would make sure, unlike the tips that I have here, which are open to the air, they would make sure that they have a cover on their tips, and they would only open the box long enough to get one tip out, and then they would close the lid of the box. So when you are working with samples in the crime lab, you would never have multiple tubes open at the same time, or tips open, or even any of your reagents open at the same time. So in the crime lab, when they're working with it, they will start with a very clean surface. They'll bleach their countertops and start with no contamination in their area. When they're adding solutions, they would unscrew the lid of the container that they want to work with. They would take the sample out, the size sample that they needed, and then they would put the lid right back on. Before they took their tube that they wanted to put it in and open the tube that they want and put that solution in the tube, and then close the lid of the tube. If they wanted to add that same solution to a second tube, they would have to go back and open the lid again and get their solution a second time from the tube. So they do not leave the lid open while they have any tubes open at all. A single thing is open at a time to avoid contamination. They will even go to the extent of a one-way flow of information in the labs. So they will have their samples, they will have their PCR, and they will perhaps have isolated DNA from a crime scene sample. Well, the crime scene sample, when it came into the lab, is never ever going to be worked on the same surface where they're PCRing the sample. So they have one room in there where they're going to examine evidence and see if there's any spots where they could potentially isolate DNA. Then that small sample is cut out and put into a small tube, perhaps, to be taken to the next lab. And only that small tube is containing isolated DNA is taken to the PCR section. Then when they have finished the PCR, they take that to the analysis area. And when it is done on the analysis, when the sizes are separated on the capillary electrophoresis, that's going to separate the PCR reaction that's has multiple bands in it now, that DNA is never ever taken back to the lab as a whole. It's a one-way flow of information. Even the paper printout that comes from the capillary electrophoresis equipment never goes back into the regular lab as a whole. So this one-way flow of information, this being careful to use sterile solutions, being careful to only open one tube at a time, being careful to wear gloves and face masks, being careful to, in everything that they do in there. All of these things serve to make sure that the proper person DNA is being isolated. They would never, for example, work on a victim and a suspect DNA at the same time because of the possibility of cross-contamination. So you can isolate multiple samples from a crime scene on one day, But then on a different day, after the surface is clean and after all your solutions are fresh, then you would work on any potential suspects. So taking great care to avoid contamination. So after you have your positive control and after you have your negative control done, and after you have set up PCR reactions in all of the different tubes, maybe you're doing 6 different PCR reactions. That's fine. You can have 1 positive, 1 negative, and 6 PCR reactions. So then you're taking them over to the PCR machine. So we have a specific program in here that is designed to have it at 94 degrees for 2 minutes. And this is because we have, as I said in lecture, that special protein blocking the active site. So this is a hot start procedure. So first we need 94 degrees for 2 minutes to get rid of that protein. Then we can have our cycles, 94 degrees, 60 degrees, and 70 degrees. This is our denature, anneal, and extension temperature. And we're doing this multiple times. And then we're allowing 60 degrees just to finish everything up, allowing the polymerase to make sure that everything that needs to be double-stranded is double-stranded. And then we're holding it at 4 degrees Celsius. So this is what the inside of the PCR machine looks like. And you simply place any of your samples, and you would have your tubes labeled as to exactly what they were. So you'd say, this is your positive control, this is your negative control. Maybe you'll have even 6 tubes in here of PCR reactions that you're doing. And you're going to put them in the heat block here. You're going to close the lid, and you're going to start the program. And you can set the volume. And in this case, if we had 25 microliters, we would want to set our volume at 25 microliters. And then we would want to run the program. And it will tell you in this PCR machine the progress that it is making. So it will say, you know, it's gone to 94, 60, 70. It will say how many times, and it will tell you how much time is remaining in the cycle so that you know approximately when to come back. Now we're going to pour an agarose gel to run our PCR samples on. After our PCR is complete, we're going to be dividing the sample into half and half. Half we're going to run on an agarose gel, and half we're going to run on an acrylamide gel. The difference between the agarose gel and the acrylamide gel is the size range which they are able to adequately separate. We have amplified 3 locations on the DNA that tend to be towards the lower side range. So we will see bands on the agarose gel. So that's a good way of saying, did my PCR work at all? But they'll all be sort of mushed together. You won't be able to see the difference between them. But on an acrylamide gel, you will see them separate out. And the longer the acrylamide gel is, the better separation you're going to see as they keep running. So we're going to make a 2% agarose gel, and a 2% agarose gel, if you remember our biochem boot camp in the beginning, a 2% of anything is 2 grams per 100 mils. So a 2% agarose gel is 2 grams of agarose. So 2 grams of agarose has been weighed out per 100 mils of TAE buffer, which is tris, EDTA, and acetic acid. So that gives it a good sodium acetate, basically, to give it a proper pH. We're going to swirl, and then we're going to heat it in the microwave. When we finish heating it in the microwave, it will be hot, and our solution will be clear because our agarose will have dissolved in the heat. While it's heating in the microwave, you want to set up your gel assembly. So this is the plate that we're going to pour our agarose into. Now, if we simply pour the agarose in like this, it's just going to fall off the side. So you need something like this, which has foam pads, which will keep the agarose in place. So you can just put it down in here. twist it so it's fixed in place. And when the agarose is a little bit cool, you don't want to pour it in when it's fresh out of the microwave. You want to pour it in when it's about 55 degrees Celsius, which means you can sort of hold it in your hand for a couple seconds before it starts to hurt. I call it the 1, 2, 3, 4, 5 ouch test. So you put it in your hand and you can go 1, 2, 3, 4, 5 before saying ouch. It's probably about 50, 55 degrees Celsius. This is because you don't want to stick a thermometer in here to actually test the temperature of the solution. We're trying not to introduce any contaminants, and a thermometer is a really good way to introduce contaminants into the solution. Alternatively, if you have a little bit more time, if you have a 55 degree Celsius water bath, you can simply place it in the 55 degree Celsius water bath and allow it to equilibrate for about a half an hour. But if you set it in the countertop and swirl it occasionally, it will actually cool a little bit faster. So it kind of depends on how much of a rush you're in. So now I'm going to go to the microwave and heat up my sample. So now our agarose has been in the microwave, and you can see it is now clear when it used to be cloudy, and it's clear because it's now dissolved in solution. When it comes back down to room temperature, it will again become a little more opaque than it is now. Before we pour it in the assembly, we have a comb, and the comb has wells in it, and the wells are designed to be placed in our holder as we pour our gel in there. And then after it is solid, when we take the comb out, we will have indentations in our gel suitable for loading our sample in. So it's basically as simple as simply pouring it all in, putting the comb in, and waiting. If you have any bubbles that you need to remove, you can use a piece of aluminum foil to drag the bubbles over to the side so that they're not in the body of your gel. So we'll come back after it's solidified. So now we're going to learn how to pour an acrylamide gel. For an acrylamide gel, because you want it to polymerize, you need to protect it from oxygen. Therefore, acrylamide gels get poured between 2 glass plates, unlike the agarose gels, which can be exposed to oxygen. For our acrylamide gel, we have a bottom spacer and 2 side spacers, which fit in here. We put a little bit of grease in this joint to keep the acrylamide from leaking out at that joint. And we also have what's going to become our spacer to make basically wells in the acrylamide solution that we are pouring so that we can have sample loaded in that position instead. So I'm just putting a little bit of grease on each side of the spacer. And I'm putting the spacer at the bottom of the gel. And I'm putting our side spacers next to it. And I'm putting the short plate on top. So now it looks like this with 2 side spacers and a bottom spacer, and we can pour acrylamide in between these 2 plates. Before you do this, you need to clamp everything down so it doesn't move. Clamp the bottom part first, because the grease will cause the spacers to spread apart, and then push down a little bit on both of the spacers. Then you can put your bottom and side clamps on. When you are ready to pour the gel, it will therefore look like this. You're going to have a wooden block to hold it up at an angle while you pour and while you make your solution. On page 119, the protocol tells you you're going to mix 48 mils of DDI water. You're going to mix 6 mils of 40% acrylamide. So here we have 40% acrylamide. And I'm pipetting out 6 mils. And then 6 mils of 10x TEB buffer. You always wear gloves when working with acrylamide because acrylamide is a neurotoxin. If you get acrylamide on your skin, it can be absorbed through your skin, and it is a cumulative neurotoxin. So the more acrylamide you are exposed to, the greater the neurotoxicity. So you always wear gloves. If you happen to get some on your skin, immediately wash the affected area. After we've done this, we need to add our APS and TEMID to begin the reaction. So now we have our solution all made. We have our 10% APS in here, and we have our TEMID in here. Remember that we've already learned that you don't want to add the APS and TEMID and then go around and talk to your friends, because while you are busy talking to your friends, this solution will be polymerizing. So you're going to add 500 microliters of APS. You're going to add 50 microliters of TEMID. Make sure everything's set up before you do both of these. You're going to swirl. And as soon as you swirl, you're going to pour. Make sure you're doing it on an absorbent pad that can absorb any mistakes that you make. So we're going to simply pour it in. We're not doing a discontinuous electrophoresis like SDS. This is all one gel. If you happen to get a bubble in there, you can simply raise it, bang it a little bit, and bubbles will rise to the surface. After you have the acrylamide all the way up, you're going to slide a comb in. You're going to slide a comb in maybe 3/4 of the way. And then you're going to clamp the comb in. If any acrylamide leaks out, you can do two things. You can fill it up with a little bit more acrylamide, which will fill the gap, and you can lay it flat. So by laying it flat, it will also stop it from leaking. So now you're going to allow it to polymerize for approximately 30 minutes, and then it will be ready to go whenever you are ready. If you needed to use it tomorrow instead of today, you would simply wrap it in moist paper towels and saran wrap, as you learned how to do in our SDS gel section. Eventually, the gel will become opaque when it has finished solidifying. You can then put it in TAE buffer, which is the buffer that we're going to use to run our agarose gel. Notice it still has the comb in it. After it has equilibrated in our buffer for 15 minutes, you can pull the comb out slowly and carefully, straight up. You don't want to do it at an angle because you want to make sure that your wells do not break. To help yourself see the wells, it's sometimes easier if you have a white piece of paper underneath. Divide your PCR sample into 2 different tubes. One tube you're going to use for an agarose gel. the other you're going to use for an acrylamide gel. Into each tube, you're going to add an appropriate amount of agarose or acrylamide dye. And then you're going to take the entire sample and load it in the lane. I'm loading it in the middle so you can see, but generally I would start on the left if I had a number of samples, and I would go to the right. So you're going to put the end of the tip in the lane, and you're going to slowly expel your solution. Then get a new tip and do the same with your second sample. Continue this until all of your samples are loaded. When all of your samples are loaded in the lane, then you can turn on the power and start running the gel. And remember, red to red, black to black. So it's going to move towards the red electrode. So we're going to swap the leads here. So it moves towards the red electrode. So it's going to run at 100 volts. The dye is going to move down the gel. Your samples are going to move down the gel. When the dye is approximately 3/4 of the way down the gel, we will have adequate separation in our samples to be able to see what we loaded in the marker lane compared with what we loaded in our PCR reaction. Then we're going to add a small amount of ethidium bromide into the buffer, mix it all in, allow it to stain for an hour. After it is stained for an hour, that ethidium bromide will have intercalated into the DNA. Once it is intercalated into the DNA, putting it on an ultraviolet light box will cause the DNA to glow so that you can visualize it. And we will show you a picture of what it looks like. Now our acrylamide gel has polymerized, and I have moved all the clamps off of the acrylamide gel, and I'm removing the bottom spacer, and I'm also removing our comb, slowly and carefully, straight up. So now we have wells that have formed that we can load our sample into. We now are ready to put it into our apparatus. We put the plate up there, and we use those same clamps now to clamp the plate in position in the gel. So now we have our gel, our plate with our gel in it, clamped into the holder that we will use to load our sample in and to electrophoresis our samples. So we're going to put buffer in. I like to put buffer in the lower container first, and that's because we took that spacer out from the bottom, and so there's a space between the glass plates. So if all you do is simply pour buffer in here straight, you're liable to get bubbles in between the glass plates. So I tilt it when I'm pouring this in, and I allow it to flow across the bottom so I don't get a bubble in between the two glass plates. And then you can put buffer in the top well, making sure if you need to that any of bubbles are removed from any of the lanes that you plan to use. If you have bubbles in lanes you don't plan to use, that's okay, they can stay. There's nothing wrong with having a bubble in a lane if you don't plan to use it. But I'm trying to get all the bubbles out just by moving them over. With a pipette, grabbing them and lifting them up. Okay, so now we have our wells, and they can be a little hard to see. If you feel you need to do that same trick by adding a little bit of the acrylamide blue buffer to each lane, the one I showed you on the SDS gels, you can certainly use that. Otherwise, we're just going to go ahead and load our samples. So you're going to come with your pipette behind the lane and place it in the well, and then slowly and carefully expel the sample into the well. When it's all in, raise it before you let go of the plunger, otherwise you'll be sucking your sample right back out. Then get a new tip with your second sample, and again, coming from behind, down into the well, loading it in, and raising it up. When you have all the samples loaded that you need, then you can connect it. And you can turn on the power. We're going to run it at 40 milliamps until we see good separation, and our dye, we have two dyes in acrylamide, a fast dye and a slow dye, until our fast dye is approximately 2/3 of the way down. And then we will take our two plates apart and we will soak it in a solution. We're just going to take this same buffer and pour it into a container and add about 1 ml of our 5 mg per ml ethidium bromide solution to that, and allow it to stain, just like we're going to stain our agarose gel. We're going to allow it to stain our acrylamide gel, and then we will put it onto the gel, put it under a piece of saran wrap on an ultraviolet light box, and we will take a picture, and you will see the result of the PCR reaction on acrylamide versus agarose.